Today we're going to talk about flow cytometry, which is an instrument that makes use of antibodies and fluorescence in order to be able to analyze single particles such as cells in very high throughput. So we're coming at it with the assumption that most of you have never heard of this technique before or don't know how it works, and so we're going to go briefly through the basics and then through some of the pros and cons. All right, so the first step is to know what the term means. Flow cytometry has three parts in the word. There's flow, which means that we have fluids moving, cyto, which means we're looking at cells usually, and then the metry, which is metric for measure. And there's two main readouts that we use for this type of technology. We look at the fluorescent intensity of labels that we may have added to the cells that we're analyzing, perhaps by using a fluorescently tagged antibody or an immunoassay on a particle, or any other type of way to fluorescently label something, such as a pH reactive dye, for example. We also measure the light that is scattered as light from a laser hits a cell or particle, and from that we can actually learn quite a bit. So for further reference, I encourage you to look at these two resources here from BD, and there's also a wide variety of other resources online if you just Google flow cytometry. All right, so what types of samples can we analyze? As I said, we're often looking at cells, and that's because we're really interested in what the instruments are built for is to analyze microscale objects, usually no smaller than a micron, although some can be designed to go down to 0.1 or 0.2 microns, and usually not much larger than 20 microns, although again, some instruments can go larger than that as long as the fluidics are designed to accommodate. Because of this size range, we're often looking at mammalian cells or bacteria, but we can also look at microparticles. For example, beads that have been coated with antibodies are very frequently used, or other structures that fall into the size range. Interestingly, there's some newer work where people actually take aqueous droplets and then surround them and do multi-layer emulsions, and then you can actually run aqueous droplets through the flow cytometer by giving them effectively a lipid layer skin. And it's important to note that you can't analyze a solid, so you have to have some kind of particulate sample that's suspended in aqueous solution. There's three parts to the flow cytometer instrument, and so it's worth stepping through each of these very briefly, and I refer you back to the BD tutorial for a more detailed introduction to each of these components. So the fluidics, the idea is to be able to move a sample of particles from one place to another, from the injection port past what we're going to cover next, which is the laser, and then out to the waste. Okay, and what's really important about the fluidics is that, as shown here, they're designed so that the cells move in single file past the point where they're going to be analyzed by the laser, so that you analyze only one cell at a time. There's another whole system, which is the optics. In this simple diagram, it's been shown as a laser, and then we can detect different fluorescent wavelengths of emission by using different detectors or different filters. But often flow cytometers have multiple lasers coming in with quite a lot going on in the optical system. And then finally, we need an electronic system so that we can convert that light that's emitted from the sample or scattered by the sample so that we can detect it with our computer. And so we're going to focus here primarily on the optics, but there's a lot of innovation that goes into designing newer and better flow cytometers by improving the fluidics and the electronics as well. All right, so what does it look like to perform a flow cytometry experiment in the lab? You have to start by collecting your sample, the cells. Those could be from cell culture, from a tissue sample. They could be isolated from blood quite commonly. And then we need to tag them in a way so that what we're interested in becomes fluorescent. In order to do that, we usually use antibodies, and because we don't want the antibodies to just bind to any FC receptors that may be present on the cells, for example, by the back end of the antibody, the FC binding to those FC receptors, we do something called blocking, where you add another antibody that binds all the FC receptors, or you add something else that will bind those. Then you can finally add your antibodies, and it's important that they be fluorescently labeled or at least tagged in a way so that you can later add a fluorophore. And as I said before, you can also use other ways to make your cells be fluorescent, such as, for example, a dye that tells you whether or not the cell is alive based on its membrane integrity. You wash out the excess antibodies, and then you finally go to flow cytometry, shown here in this step, where the cells are injected and then pass single file past one or more lasers in order to make detection. And then we analyze this multi-parametric data where, say, we're analyzing blue fluorescence on the x-axis and red fluorescence on this y-axis, and we can tell whether a cell is bright in one or both of those dimensions. When it comes to analyzing data, we're thinking here about potentially many fluorochromes. As many as 12 to 14 are routinely collected now by high-end spectrometers, and we can even now go up to over 20 with the top flow cytometers. So then we're stuck trying to reduce that usually into at most a two-dimensional plot. And so there's a lot of clever thinking and innovation that goes into how to analyze such a multi-parametric system with classic plots that people are used to. But in terms of display, we often use a histogram, such as shown here, where the intensity of the one species is on the x-axis, and then the number of cells that had that intensity are on the y-axis. So in this case, a peak on the right side of the histogram means that those cells were brighter, and then this peak here on the left side, those cells were dimmer. If you want to plot two parameters at a time, you would use usually a scatter plot or a variation thereof, such as shown here, where two parameters, in this case a forward and side scatter, are being plotted against one another in an XY format. And so if you're up here in the upper right quadrant, it means that they're both bright, that the cells are bright in both of those parameters. If you're down here, it means the cells are dim in both, and then you can figure out from there. And then there's variations on the XY scatter plot that you've seen in other data display formats, such as a contour plot or a density plot with color. So any way that you can find to convey the information that is present in this multi-parametric data. All right, when it comes to choosing fluorochromes, this is non-trivial, particularly as you get to multicolor fluorescence with the analysis. It really matters that you choose the right fluorochromes, and the things that you need to think about are, first, what instrument are you working with, and therefore what fluorescent filters do you have available and coming off of which excitation sources, usually a series of lasers. So you need to choose fluorochromes that can be excited by the lasers and then detected through those fluorescent channels. But you want to minimize what we call spectral overlap, which is shown here in this plot of several classic or very common fluorochromes. And you can see that here we're just plotting the emission spectra. They overlap quite a bit. And so if you have an emission filter that's trying to detect, for example, just this Alexa Fluor 647, if the bandwidth of that filter is wide enough, you're going to pick up bleed over from the Alexa Fluor 700 or from this dye over here, for example, which is PE size 7 that has like a short wavelength tail as well. And so you have to think carefully to minimize that overlap and try to choose fluorochromes that have minimal overlap in your filters. Finally, there's an even more subtle aspect of choosing fluorochromes, and that's to account for the fact that some of the antigens that you're going to be detecting on the surface of the cells might be present in high abundance on those cells, and others will be very rare. And so in general, you want to put your brightest fluorophores on the markers that are the least abundant. That way you're bound to get the most signal. If you put a very bright fluorophore on a highly abundant target, then you get so much signal you might saturate the detector and not be able to resolve other markers that have lower abundance. All right, so what are the advantages? Why do people like flow cytometry so much for particular applications? The first main thing is that this gives you single cell data, which means you get the full array of every single fluorescent channel and the scatter for every single cell. So you can imagine these are very large data sets with a lot of information, and you can do it for a lot of cells very quickly. So many flow cytometers will analyze many thousands of cells per second, and it's very common to say that you will always collect at least 5,000 or 10,000 events or cells for every sample, and so you're getting a lot of data here. The other benefit is that because it is easy to multiplex by having these multiple fluorescent lasers and multiple filter sets, it's really easy to multiplex multiple markers. And so again, this rich data set for multiple markers across every single cell and a high number of cells. Now, it's not all good news. There are, of course, some downsides to this technique, just like any other technique. For example, because of its sophistication of the instrument with the fluidics and the optics, it requires a lot of training to perform this type of analysis so that you don't accidentally clog the fluidic lines with samples that have clusters of material in them or a bulky matrix element, for example. You can accidentally choose a mismatch of fluorophore to the optical system that you have at hand, and then you won't get good signal. And you have to learn how to analyze this sophisticated data. So, for example, at UVA, the flow cytometry facility offers a course that's an entire week with both lecture and lab before anyone is permitted to use the instruments. You also have to prep your samples correctly, in particular to avoid aggregates so that you don't clog the laser lines. So a sample that has large aggregates in it must either be filtered or can't be used on a flow cytometer. In terms of choosing what to analyze, you are limited to, say, a dozen or perhaps at very most 2 dozen colors because of the spectral overlap, although that is getting better and better as people invent fluorochromes with narrower excitation and emission spectra. There's a major element of cost because the antibodies that are usually used for flow cytometry are very expensive and also can suffer from reproducibility issues between vendors and even between lots from the same vendor. Validation of your antibodies becomes really critical. And finally, the data here is usually qualitative, meaning we can tell if a cell has high levels of a particular antigen or low levels, but it's very unusual to use this to quantify, say, the concentration of a protein on the surface of a cell. It is possible to do that, but it's usually not done because of the nonlinearity of the fluorescence measurement under typical conditions. All right, so that's it for today. After this, we'll be ready to do an activity for flow cytometry to learn more about how to choose color panels and how the instruments are designed. And then that's it. So now you should be able to at least be comfortable seeing flow cytometry appear in a paper and ready to interpret the data that you see.